From my research findings, I am going to present on gene expression, transcription, and translation. So what is gene expression, transcription, and translation? Gene transcript expression is the process by which genetic information is encoded in DNA and is used to produce functional proteins that are essential for the proper functioning of cells and organisms. Transcription is the first step of gene expression. So this takes place in the nucleus of the eukaryotic cells. It involves the conversion of DNA into messenger RNA, which carries the genetic instructions for the DNA to the ribosome in the cytoplasm. Translation is the second step that occurs on ribosome in the cytoplasm, and it involves the coding of the mRNA to assemble amino acids in a polypeptide chain that form a functioning protein. So transcription initiation. Transcription initiation is the first crucial step in the process of gene expression. It begins with the binding of transcription factors to a specific DNA sequence that is called the promoter region that is located before the gene that needs to be transcribed. Transcription factors are proteins that help regulate gene expression by facilitating or hindering the binding of RNA polymers to the enzymes responsible for creating the mRNA molecule. When the appropriate transcription factors recognize and attach to the promoter region, they act as signals for the assembly of RNA polymers 2 at the transcription start site. So this event makes the initiation of transcription and the collaboration between transcription factors and RNA polymers to form a transcription initiation complex that paves the way for the subsequent stages of transcription. So transcription elongation. So what is transcription elongation? It is the process of synthesizing the messenger RNA molecule that continues as RNA polymers 2 and moves along the DNA template strand. So this occurs when the initiation phase, where the transcription factors have binded to the promoter region and RNA polymers 2 and has unwrapped the DNA double helix. So during elongation, RNA polymers adds contemporary nucleotides to the growing mRNA chain one at a time, and the nucleotides are based on the DNA template strand. So transcription termination. Transcription termination is the final stage of the transcription process in eukaryotic cells. It occurs when RNA polymerase 2 reaches a specific DNA sequence that is called the polydination signal. This signal serves as a termination signal for the RNA polymerase 2 and marks the end of transcription for the gene that was being transcribed. So basically, when RNA polymerase 2 encounters that signal, it prompts the addition of a series of adenine nucleotides to the 3' end of the mRNA molecule, and this results in the formation of a polyatyl, and the polyatyl plays a crucial role in stabilizing the mRNA and protecting it from degradation by cellular enzyme. The next step is the step of making the proteins. This means that there are several steps to actually configure the protein, so it would have to go through post-transcriptional modifications. So after the transcription, the freshly synthesized mRNA molecule will undergo a series of crucial modifications before it is ready for the translation in a functional to be ready in a functional protein. So these are known as, post-transcriptional modifications, but they play a role in ensuring stability, proper functioning, and efficient transport for mRNA from the molecules to the cytoplasm. So these post-transcriptional modifications collectively ensure the integrity and proper functioning of the mRNA during its journey from the nucleus to the ribosome in the cytoplasm. The modified mRNA will now be equipped to withstand the cellular environment and will be then ready to undergo translation. The first process would be translation initiation. Translation initiation is the first step of the protein synthesis. The mRNA molecule will bind to the small subunit of the ribosome in the cytoplasm. Along with initiation factors, the small ribosome subunit then locates and will attach to the mRNA start codon. Then translation elongation will happen, so the ribosome will move along the mRNA from the 5' to the 3' direction, and the large ribosomal subunit joints of the initiation complex and the initiator tRNA carrying the methionine will be positioned in the ribosome's P site. The A site of the ribosome will now be changed to the next charged tRNA. So each tRNA molecule will bring a specific amino acid that has an anticodon that will pair with the mRNA codon in a contemporary manner. So the ribosome catalyzes the formation of a peptide bond between the methionine and the amino acid and carried by the tRNA in the A site. So then translation termination happens. This culminates during the termination stage when a stop codon is reached out of the mRNA. So stop codons such as UAA, UAG, or UGA do not code for any amino acids. Instead, they will release factors that bind to stop the codon, triggering hydrolysis of the bond and hydrolysis between the bond and the polypeptide chain and the tRNA in the P site. So this will lead to the release of newly synthesized proteins from the ribosomes, and subsequently the ribosomes will dissociate into two subunits and the process of translation will be complete. So next will be the actual protein. So protein synthesis is a process essential for the functioning of all living organisms. So proteins will play diverse roles in cellular functions, including acting as enzymes that catalyze biochemical reactions and signal transduction pathways. So the accurate synthesis of proteins is crucial for being able to maintain normal cellular activities and overall organismal health.